Hello, everyone, and welcome to today's webinar titled High-Throughput Assay for Monitoring COVID-19 Antibodies and Their Isotypes. This webinar is part of an ongoing coronavirus virtual webinar series. I'm Susie Valdez of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and sponsored by Luminex. For more information about Luminex, go to luminexcorp.com. Now let's get started. Before we begin, I want to remind everyone that this event is interactive. We encourage you to participate and submit as many questions as you'd like during their presentation. To do so, simply type into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. If you have any trouble seeing or hearing the presentation, click on the Ask a Question box located on the far left of your screen. As a reminder, this presentation is educational and offers continuing education credits. Click on the Continuing Education Credits tab located in the top corner of your presentation window and follow the process to obtain those credits. I now present today's speaker, Dr. Susan Zolla-Pazner, Professor of Medicine at the Division of Infectious Diseases at Icahn School of Medicine at Mount Sinai in New York City. Welcome, Dr. Zolla-Pazner.
Thank you, Susie. Today, I'd like to share with you the work that we've done over the past 2.5 months to develop a high-throughput assay for antibodies specific for SARS-CoV-2 in COVID-19 patients, and show you some of the results that we have achieved using this assay. So just as an introduction, though you may not need it, I just want to share with you this cartoon of the structure of the virus. We're going to be focusing today on the spike protein, shown in yellow, that covers the surface of the virus particle. Sometimes antibody assays are directed toward the nucleocapsid, which is shown in pink. But the work that we are doing today is focusing on detecting antibodies to the spike protein and a portion of the spike protein, which I will show you now. So rather than a cartoon, this is a molecular diagram or molecular structure of the spike protein as a whole, with the receptor binding domain highlighted in red. In this slide, what you can see is that the spike protein is anchored in the membrane of the virus envelope, which is at the bottom of the slide, or would be at the bottom of the slide. And then the upper part of the structure, where the receptor binding domain resides, is the distal portion of the S protein. Now, in terms of a description of the antigens that we use in the assay, we use the spike protein, as I said, and the receptor binding domain, which is abbreviated as RBD. Let's go over the details of the spike protein. It is referred to, or it comes from the S protein. It is constituted of the S protein. And the spike itself is a trimer, a homotrimer of 3 copies of this S protein, each of which is approximately 140 kD. The spike protein binds to the host cell via the surface angiotensin converting enzyme 2, ACE. So the spike protein binds to the ACE2 on the surface of receptive cells. For coating the beads that we use in the assay, we use the S protein ectodomain, which is composed of 1,213 amino acids. And at the C-terminus of the S protein, a thrombin cleavage site has been added along with a T4 fold-on trimerization domain and a hexahistidine tag. This was designed and expressed by Florian Kramer at Mount Sinai School of Medicine. Now, a portion of the spike protein that I showed you before in red in the previous slide is the receptor binding domain. This is an area of the S protein that consists of amino acids 319 to 541. Out of this protein that I mentioned to you has 1,213 amino acids. So this is in the middle. This is a region in the middle of the protein. And this antigen, as we're using it, consists of the natural N-terminal signal peptide of the S protein fused to the RBD sequence and then joined with the C-terminal hexahistidine tag at the C-terminus. The histidine tags are used for purification of the proteins as they're expressed. But just a quick summary, introduction and summary to the Luminex assay. The principle of the assay is that there are different bead sets here labeled A4, A5, 6, and 7. And each bead has an individual fluorochrome tags that identifies each of the different bead sets. In the assay I'm going to be talking about today, we're only using 2 bead sets, one that binds to the S protein, to the spike, and the second bead set that binds to the RBD protein. We mix these antigen-coated beads with the serum that contains the antibodies that we're looking for, or in the case of the controls, of course, there is no antibody there. That's what we're trying to distinguish. So we have the beads. You see down at the bottom that the antigens are either the S protein or the RBD, and then we add the serum or plasma. If there are antibodies in the serum or plasma, it coats the bead. It binds to the coated bead. That's shown in the upper right here. So you have the antibodies bound to the antigen, bound to the bead. And then we use a biotinylated detection antibody, which we call a secondary antibody. In this case, it would be one of a number of different types of anti-immunoglobulins. So a secondary antibody that binds all immunoglobulins, or one, for instance, that only binds to IgM or only binds to IgA, et cetera. And we'll go through that in a minute. And then for the readout, a final reagent is added, which is streptavidin labeled with phycoerythrin. It will bind to the secondary antibodies and the biotinylated secondary antibodies. So as I mentioned, the detection is with secondary antibodies to total IgG, total IG, to IgG subclasses, IgA subclasses, or to IgM. These beads, once they are coated, mixed with the plasma specimen, mixed with the secondary antibody, and mixed with the streptavidin phycoerythrin, are then added into 96 well plates. Both bead sets are in each well. And then the contents of the wells are read in a Luminex analyzer, and we can determine if there are antibodies that are binding to RBD and or the S protein. We can quantitate that with a titration curve, and I will show you that in a minute. This is the outline of the assay. We can coat the antigen onto the beads and then store the beads for at least a month at minus 80 degrees centigrade, and then use them as we need them. We add the washed coupled beads and the serum to wells in the 96 well plates. We wait 30 minutes, and then we add the biotinylated secondary antibody, the anti-Ig. We wait another 30 minutes. Of course, there are washing steps between each of these. And then we add the streptavidin phycoerythrin, wait another 30 minutes, wash, and then read in the Luminex instrument. So the assay that I'm going to show you is both qualitative or binary assay and a quantitative assay. And I'm going to show you some of the data that we have generated now. So this is a dot plot of the screening of plasma from COVID positive and negative subjects. On the left, we're showing the results with the beads that are coated with the spike protein. M just stands for the fact that it was expressed in mammalian cells. And on the right, you see the results using the RBD, also expressed in mammalian cells. These data were generated with the plasma that were diluted 1 to 200. And as you can see here in red in both graphs, the COVID-19 positive patients score strongly, and the COVID negative patients have only background reactivity. This is an experiment that was done using plasma from volunteers. And in the next slide, I'm showing you the data with sera. So you can use both serum and plasma, and they give equivalent results. What I'm showing you in this slide, again on the left, is the reactivity of the sera to M spike, and on the right is the reactivity to RBD. Again, the sera are being tested at a dilution of 1 to 200. On the right of each side are 3 negatives, COVID negative controls. And then the colored bars represent COVID positive subjects. Now, there's some, the first thing that you need to notice here is that it's obvious that amongst the COVID positive specimens, there are tremendous differences in the levels of antibodies. And this we see commonly. The levels vary enormously. What has been reported in the literature is that patients with severe disease have a higher level of antibodies than patients or subjects who've had much milder disease. And I can show you that in just using these few samples amongst many others that we've done. The 3 bars that are outlined in black here, that are shown, the bars are in red, show the sera from a single patient taken at day 8, 11, and 15 after the beginning of symptoms. And as we know, after an infection, one of the things that you can use to diagnose an infection is an increasing titer of antibodies in the serum, and that's exactly what you see. And you can see that in a very short time, in a period of 1 week, from 8 days to 15 days, as is shown over these 3 bars, the level of antibody is going up dramatically, both to spike and to RBD. In this slide, the orange bars are highlighted, and these are longitudinal specimens taken from a different patient. And these 2 specimens were taken at days 7 and 11. And again, you can see the rising titers. The other bars represent a variety of patients or a variety of specimens from individuals who had tested positive in the clinical lab. so their PCRs were positive, and their antibody levels are all positive, well above the cutoff for negatives, which are so low that you can't see them. It's not that they weren't done; they were done, but the levels were so low that you don't see the bars for the negatives. So all of the positives are positive for the presence of antibodies. We ran these 6 against spike, and then 2 of these 6 were run against the RBD, show similar results. Now, these are what we call the results from screening. We screen the sera at 1 to 200, and those specimens that are positive at 1 to 200, those specimens that are positive for RBD are titrated. And what I want to show you in this slide are the titration results with the same specimens that I showed you in the screen in the previous slides. So you can see these beautiful titration curves that we get when we're titrating from 1 to 50 out to a dilution of just over 6,000 here. You can see that at it's 1 to 6,400, we're down close to baseline with most of the specimens, but some of them still have quite high titer and will probably go out to 12,000 or even 24,000 in this particular assay. When we do have in negatives in this graph shown as N1, 2, and 3 in black, they lie right on the y-axis at 0. But to give you a better sense of the negatives, I'm showing you in this slide what the negatives look like in terms of the titration. The green lines lie right at the very close to the 0 mark, whereas a positive pool of sera at 1 to 100 dilution scores appear where the red dots are. And again, the negative pool and the PBS between negative controls are in black. And again, it's very hard to see them because they're right down at baseline. We also ran a series of specimens that were drawn from volunteers in an IRB-approved study. They're pre-pandemic sera that were drawn before November over, I think, 2017 to 2019. On the right, we're showing 2 positive sera and the red bars. The next 2 tiny bars were negative sera. Over on the right-hand side are specimens that come from the positive pool that I showed you before, this very high blue bar, the negative pool, and the PBS. And the pre-pandemic specimens, you can see, range all the way in the middle here. They are just barely detectable with the exception of this last specimen, which was actually negative in the RBD screen, but did come up slightly positive in the spike. So now what I'd like to highlight is the relative properties of the ELISA assay and ELISA assay versus the Luminex platform. So in this slide, we titrated the same serum, and this is from that first patient where we had the longitudinal specimens at day 8, 11, and 15. They're color-coded here, so this is the specimen from day 8 in sort of salmon color. In red is the specimen from day 11, and in maroon is a specimen from day 15. And in the upper graph, we're looking at the results generated with the Luminex. And in the lower graph, you see the results generated by ELISA. And what you can see by looking at the 50% half maximal binding value, which is identified by the dashed lines, is that the half max value or titer that we identify, for instance, in the 15-day sample by Luminex is 1 to 2,500, whereas the same specimen in ELISA has a half maximal value of less than 1 to 500. So there is at least a five-fold, and in some cases up to a 10-fold difference with the Luminex assay of being much more sensitive. In addition to the greater sensitivity of the Luminex assay, we have been able to reduce the amount of antigen needed per test. So with the ELISA, the wells are typically coated with 0.05 milliliters or 50 microliters of the antigen solution at 2 micrograms of antigen per ml, and using that in 96 well plates means that you're using 0.1 micrograms of antigen per test. In contrast to that, when we can coat the Luminex beads with 12 micrograms of antigen per 3 million beads, which gives us 1,200 tests. And that means that we can use 0.005 micrograms of antigen per test. So a 20-fold decrease in the amount of antigen that you use, which obviously results in cost savings. So another advantage of Luminex over the ELISA assay is indicated here. In the Luminex assay, we can couple the beads in advance and store them frozen until we need them. Once we need them, we take them out of the freezer. We incubate them with serum or plasma for 30 minutes. We add the biotinylated secondary antibody for 30 minutes, and then we add the streptavidin phycoerythrin for 30 minutes, and then we read. You'd have to add a few minutes here for each of the washes, but generally this test takes only an hour and a half to do. And you do the RBD analysis and the spike protein analysis simultaneously. So within an hour and a half or 2 hours, you have the information on 28 specimens in the plate in just under 2 hours. In contrast, in the ELISA plates, you have to coat the plates overnight. Then you have to block for an hour. Then you have to add serum for 2 hours. Then you add horseradish peroxidase secondary antibody, then for another hour, and then the substrate before you can stop and read the plates. So the ELISA requires an overnight preparation of the plates and then a 4-hour assay to do 14 specimens. Moreover, you have to do the RBD on one day, determine which ones of those are positive, and then do the spike on the following day. And so that means that the turnaround time is 2 days for the ELISA test, whereas the turnaround time for the Luminex is just over 2 hours. So 2 hours versus 2 days. I can summarize the advantages of the Luminex over the ELISA as is shown here, which I've already mentioned. It provides both a qualitative or binary assessment and a quantitative platform for the assessment of COVID-19 antibodies. It uses 20-fold less antigen per test than the ELISA. It is at least 2.5-fold faster to run, actually even faster than that. It can run in a half a day or 2 hours. Half a day if you include the time it takes to dilute the samples, but if you have a robotic system, that is done for you. But it can run in less than 1 day. In addition, we have determined, although I'm not showing you the data, that you can run the assays on 1 day and read them on the next day if that is more convenient in the context of the lab that's doing the assay. We can perform the RBD screen and the spike titration simultaneously with Luminex, but with ELISA, it's a sequential assay for the RBD and then the spike. As I mentioned before, the turnaround time is much faster, and the level of sensitivity is 5 to 10-fold better in the Luminex than it is in ELISA. Both serum and plasma can be used, and that's true for the Luminex as well as the ELISA. Now, that is a description of the antibody test for presence and level of total antibodies in serum and plasma. But we were particularly interested in looking at what types of antibodies are produced in response to infection with SARS-CoV-2. And so I'm going to show you a series of experiments now looking at the different types, isotypes of antibodies that are in the serum and plasma of patients. So here is the method. It is basically the same method that we used before, but we use antibodies to the various subclasses of, or isotypes of antibodies, and I'll show you that in a minute. How did we pick the antibodies that we were going to use as secondary antibodies? A lot of secondary antibodies or anti-immunoglobulins are sold by companies that are labeled as being specific for IgG1, IgA, IgM, etc. But in fact, when you test them, they are not necessarily as specific as they are labeled by some of the companies. So we wanted to be sure that what we were measuring when we said we're measuring IgA is truly IgA, and that the secondary antibodies are specific for IgA and not cross-reactive with other isotypes. And so we did a control experiment that I'm going to show you. And for this control experiment, the beads were coated with myeloma proteins of known immunoglobulin isotypes. So here, for this control, only for these control experiments, we coated beads with myeloma proteins of IgG1 or IgA or IgM, etc. And then these beads were incubated with the various secondary biotinylated antibodies that we bought from various companies that were labeled as being specific for total Ig or for the IgG subclass or the IgA subclass or the IgM. And then we developed them as we normally did with the streptavidin. And in this slide, you can see just some of the reagents that we tested and that we decided to go forward with. I'm not going to show you the secondary antibodies that didn't work because we don't use them. There is an antibody that we use that measures total Ig, total immunoglobulin, and that secondary antibody reacts with beads coated with IgG1, myeloma proteins, or IgG2A, IgG3, IgG4, IgA1, IgA2, and IgM. That's in the upper left-hand corner. On the upper right-hand corner, you see the isotypes that are picked up by a reagent, a secondary reagent, that is specific for IgG. So just a minute ago, I showed you a reagent that picks up total immunoglobulin, which is GA and M. But on the upper right, you see the controls with the secondary antibody that picks up IgG1, IgG2, IgG3, and IgG4, but not IgA1, A2, or M. Another excellent reagent was specific for IgA1 subclass. This is on the lower left, and you see that it only reacts with IgA1 myeloma protein and none of the other myeloma proteins. And similarly for the antibody to the IgM subclass, in the lower right, you see that this antibody reacts with the IgM coded beads. There is a little bit of cross-reactivity with IgG3 and IG4, but it's much less, and we decided we could live with that for the IgM-specific antibody. So using reagents that were designed and selected for their specificity in the Luminex assay, we went on to analyze the isotypes in serum and plasma of the SARS-CoV-2 infected patients and in uninfected donors. So in this slide, what I'm showing you is the level of total immunoglobulin against the M spike. And this is in sera and plasma. So this is looking at total Ig, which means IgG, IgA, and IgM. This is looking at antibodies specific for the spike. On the right-hand side, you can see that there's very, very little reactivity, just background activity, defined by the negative specimens that we tested, many of which came from the blood bank. The dotted line that is hard to see because it is so close to the baseline is the cutoff, which is based on the mean plus 3 standard deviations of the negative specimens that we run in each experiment. And what you can see here is that total IgG is found in all of the positive specimens. As I pointed out before, the levels vary tremendously. The readout, as with all of these experiments, was with mean fluorescence intensity, which is the way the Luminex instrumentation reads out the light units. So all positive patients were positive for antibody to spike protein. All the negatives that we tested were negative. This is a slide that shows you levels of IgG-specific anti-spike antibodies. And this was a surprise to us because there were many of the COVID-19 patients that are shown in the left 2/3 of this graph that had very good titers or levels of IgG, but there were several that had very low levels or no levels of IgG. This was quite surprising. We expected that they would all have IgG, although I have to stress that most of these specimens come from individuals who are donors to the program at Mount Sinai to provide convalescent plasma for use as treatment. And they are, the titers of their antibodies are tested, and then if they have good antibody levels, they are selected for plasma donors for convalescent serum. We have no information about how soon after infection or how soon after the end of symptoms these patients or these subjects are coming in for testing. So many of these may be relatively early after infection and don't seem to have IgG antibodies. In contrast, and these slides that I'm showing you are all the same patients. In contrast, if you look at the IgA from these same individuals, all of them, with the exception of one individual, have IgA antibodies. Again, the baseline is this dotted line. You can just barely see it above the baseline over on the right when you're looking at the normal donors. So all but one of these individuals had IgA antibodies. And in the next slide are the IgM antibody levels in these individuals. And in fact, every single one of the individuals who we tested had IgM antibodies. Again, the levels vary, but all of them had IgM. So if you look at the percentage of responders, meaning individuals who are COVID infected and have antibodies to spike, if you're measuring total Ig, total immunoglobulins, which includes IgG, IgA, and IgM, 100% of them have antibodies, which you would expect. But a minority of them have IgG antibodies, 61%. 92% had IgA, and 100% had IgM. So of the 33 specimens that were tested in this particular series of experiments, from 31 patients. The patients that we looked at were actively ill, or the majority of them were convalescent, and the IgA and the IgM response rates were higher than the IgG response rates, which was not expected. Now, you cannot compare the levels of antibodies, isotypes, using different anti-IgG, IgA, and IgM. For example, if you use an anti-IgM secondary antibody, you can look at all of the patients, as we did here, and we can say that the patient with the highest spike had, relative to other patients, was the most robust responder in terms of IgM. But the level that you read probably is quantitatively different than you would get with an antibody against, say, IgG1. And I can show you this on the basis of the y-axis. So the y-axis here, read out in mean fluorescence intensity, goes up to 300,000. Whereas when you're using an anti-IgA antibody, the axis really only goes up to about 40,000. And if you're looking with anti-IgG, you only go up to about 175,000. So you can compare between patients with the same isotype, but you cannot compare levels within a patient from one isotype to the next. So in order to understand better how to analyze the data, what we did was take each one of these graphs and drew it into quartiles. So for the IgM graph, for instance, we counted up the number of specimens that had between cutoff and 50,000 MFI, and we said that that was the number of specimens that were in the first quartile. And then the second quartile was between 50,000 and 100,000, and we counted the number of specimens that fell between 50 and 100,000. And then the third quartile was between 100 and 150 MFIs, and then the fourth quartile was anything over the 150,000 MFI. And so we could look at patients and say, okay, is this patient, let's say the first bar, is in the first quartile for IgM, but is perhaps in a higher quartile in the second quartile for IgA, and is, let's see, and is in, say, the second or just barely into the third quartile for total Ig. And using quartiles, we can compare quantitatively the amount of the different isotypes in the different patients. So what I'm showing you here and on the next slide is the analysis in terms of the quartile. So for instance, if you look at patient P number 4, this patient specimen is in the third quartile. That's the next to the highest quartile for Ig, total Ig, that's GANM. But this specimen didn't have any IgG in it. It was in a low quartile for IgA, but it was in a very high quartile for IgM. So it means that at this point in time, patient number 4 had mainly an IgM response. If you look down on the bottom row at patient number 7, TF number 7, you can see that the majority of the response in this patient is IgA, and there's very little IgM, and there's a medium amount of IgG. If you look overall at this graph, at this table, what you see is what I showed you before, that if you look at it, it's very clear that the positives, the strongest positives are in the IgA and IgM classes, whereas many fewer of the individuals have strong IgG responses. The patients at the top here, 1A, 1B, 1C, 2A, and 2B, who we talked about before, were acutely ill, severely ill, and they are the ones that have the highest antibody titers and levels, and that is already established in the literature. For the rest of the specimens that I showed you in the graphs above, you see, again, if you look at TF number 9 in the second row, you can see that the major response is IgA. If you look at TF number 12, number 11, the major response is IgM, etc. Another way of seeing this, the fact that different patients have different relative levels and different relative patterns of isotypes. You can look at these two graphs, which are the graphs I showed you before for IgA and IgM. And I've drawn these two-headed arrows connecting two of the patients, showing you that the first arrow, that this particular patient had a great deal of IgM, but very little IgA. Had very little IgM, but a great deal of IgA. And the second arrow shows just the opposite, patient that has a high amount of IgM and very little IgA. So different patients have different patterns of responses in different isotype categories. And one of the things that is important to figure out is what accounts for these differences in responses. What is the utility of isotyping these antibodies? The first is to determine if the presence of different isotypes affects the therapeutic potential of transfused convalescent plasma. So right now, the convalescent plasma that's being transfused into severely ill patients is chosen on the basis of titer alone. But it might be better, we don't know, but it might be better, it might be more effective if you chose units of plasma that had high titers of IgM, for example. And we are looking into that, looking at and asking questions like this in conjunction with the transfusion team at Mount Sinai. The second question that may be answered by these isotyping studies is to determine if antibody levels and isotypes change over time in convalescent plasma, and if this affects which plasma donors should be chosen. So you would expect, probably, that the isotype patterns will change over time, and that could imply that some plasma donors who seem to be good plasma donors early after they have recovered from infection, even if their titers stay up, the isotype pattern may have changed later during recovery. And it might be important to determine if the isotypes in this serum or plasma from these individuals, say 6 months after recovery, is different than the plasma that was derived for transfusion from these patients, say 1 month after recovery. And finally, it's very important to understand the nature of the antiviral antibody response, the relative protective activities of the different antibody isotypes, and the immunopathogenesis that might be induced by antibodies that have been produced in response to the virus. Finally, I want to talk about the reproducibility of the assay. I'm showing you in this slide the correlations between two anti-spike antibody experiments. And you can see the very good correlations. The R values are above 0.9. The P values are less than 0.0001 for each of the isotypes that we were looking at. So it's very reproducible assay to assay. And you would expect that there would be a correlation in the presence and levels of antibodies to spike and to RBD. And that also is true. As you can see here, all of the p-values are less than 0.0001. Finally, let me sum up what I have told you in today's webinar. We have developed a Luminex-based assay for COVID-19 antibodies, which is both qualitative or binary, and it is quantitative. The advantages of the Luminex assay versus the ELISA assay is that it uses 20-fold less antigen per test. It can be run at least 2.5-fold faster than the ELISA itself is run. The turnaround time is less than a day versus 2 days for the ELISA. You can perform the RBD screen and the spike titration simultaneously, whereas with ELISA, it has to be done sequentially. And the level of sensitivity is 5 to 10-fold better for Luminex than for ELISA. And this results in fewer false negatives. The isotypes of COVID-19 antibody in serum and plasma can be analyzed and quantitated. The relative patterns of IgG, IgA, and IgM differ from patient to patient, and the IgA and IgM response rates were higher than the IgG response rates, at least during the period of time from which the specimens were collected at this relatively early stage of the pandemic. And the sensitivity and specificity of the assay using RBD and spike proteins is quite high. In conclusion, I would like to acknowledge the fantastic people who I've worked with, wonderful scientists and wonderful collaborators, in particular Svenja Weiss and Jeremy Klingler, who are postdoctoral fellows in the lab, and Dr. Katarina Hugh, who is a principal investigator who I've worked closely with for many, many years. So I thank them especially and all of the people who are listed here. I show my contact information down below in case you would like any further information about the assay that we have developed. And thank you very much, Susie. I'm ready to take any questions that may come up.
And thank you, Dr. Zolla-Pazner, for that informative presentation. And we will now start that Q&A portion of our webinar. If you have any questions you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. So let's get started. We have quite a few questions already coming in, Dr. Zolla-Pazner. Our first question from our audience member is, what dilution of the serum is used in the assay?
So for the screen, we have been using the 1 to 200 dilution. For the titrations, we usually go from 1 to 50 out to about 1 to 10,000. But it's sufficient to screen at 1 to 200.
Thank you for that. And do you know if the pre-pandemic positive signals you reported were a result of the cross-reactivity with another coronavirus infection or COVID-19? In other words, was there a respiratory pathogen panel performed?
No, we did not do a respiratory disease panel, but Dr. Florian Kramer did. I believe that is published in his paper, which was in Nature Medicine about a month ago, and he did not find any cross-reactivity between COVID-19 and other coronaviruses. Of the 50 specimens that we looked at, only one of them had a positive signal. They all should have been negative. One of them was positive against spike and negative against RBD. We don't have any clinical information on that particular patient.
Thank you for that. And do you see the difference in using serum versus plasma in the AB test? And it's a two-part question, actually. Is there an impact of AB, titers in using the lipemic and hemolyzed samples? I apologize if I mispronounced that.
Okay, so we don't see any difference in looking at serum or plasma. I say that because our values are very similar for the panels of serum and plasma that we're using. Although we have not tested in parallel serum and plasma from the same donor at the same time point. But we're not seeing any significant differences between serum and plasma results. And so we are concluding that you can use either one in your assay. I am not sure about the question of whether hemolysis impacts the results. My impression is that it doesn't, because some of the assays that we have done, and I didn't show you those data in my presentation, but we have been working with some specimens that are hemolyzed, and we're still getting good reads with them. So again, I don't have a specimen that comes from the same donor, the same bleed that is hemolyzed and is not. But my impression is that if the antibodies are there, the hemolysis does not seem to create an artifact.
Thank you for that. And I want to thank our audience for these great questions that are coming in. We have time for a couple of more. This next audience member says, very nice presentation. Thank you so much. Tetracore has FlexM array SARS-CoV-2 human IgG assay for Luminex platform, and we validated our assay and submitted EUA application. They want to know if you'd be possibly interested in comparing the assay with yours.
Absolutely.
There you go, right? And for the coupling, what's that?
My contact information was at the bottom of the last slide. I don't know if you can still see it or not, but you can look me up on the Mount Sinai website and you'll find it.
Wonderful. Always great how people can get the collaborative opportunities from these presentations. For the coupling of the S antigen to beads, did you find that you needed to use the Avidin beads, or did you use the standard MagPix microspheres?
I'm not familiar with the alternative. I know that we have used the beads come from Luminex, and I don't remember the exact model number, but I can provide that information to you offline if you send me an email.
Thank you so much. We have time for one more question. Can you please comment on the stability of the Luminex assays when you freeze versus storage at 4 degrees?
You know, it really depends on the antigen that you're using. So recombinant antigens can be fussy and can go bad. Of course, we've only had a couple of months to be dealing with the spike protein and RBD, and we have not had any difficulty with them. We usually store our coated beads, especially if we make large amounts of them, at -80. And then if there's any left over, we will keep them at 4 degrees and use them for the rest of that week, but never longer than that.
Thank you for that. And just, I want to say, Dr. Zolla-Pazner, thank you for this presentation. Did you want to provide any closing remarks for our audience before we leave today?
I just think that it's important to point out that with the scale up for mass screening that is really going to be required nationally and internationally, that this platform provides the ability to scale up in a much more efficient way than using the ELISA format. So this is an assay design that would best be suited for reference labs where you're doing thousands of tests a day, and that is what it was designed for.
Absolutely. And thank you again for this incredible presentation and for your important research. And before we go, I want to thank the audience for joining us today and for their interesting questions. I'd also like to thank our sponsors, Luminex, for sponsoring today's webinar. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. And this webcast can be viewed on demand through the end of the year 2020. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's event. Until next time, have a great day and be safe. Bye-bye.
Thank you.